Hi everyone, thanks for joining the Vaccines 2020 virtual meeting. My name is Xiangsu from Microfluidics. I would also like to thank the meeting organizer for hosting this great event and give me the opportunity to talk about our solutions for vaccine components manufacturing, especially for large-scale productions. I'd like to start with a quick introduction about who we are and what we do for those of you not familiar with Microfluidics, and then talk briefly about vaccine technologies and their main components. And then hopefully I can explain why the Microfluidizer technology is unique and is the most suitable technology for manufacturing some of the vaccine components by demonstrating you with two case studies. The first case study will be producing lipid nanoparticles for antigen delivery. The second case study is a case scale-up study for vaccine adjuvant nanoemulsion production. I will then show you our product line and end my presentation and be ready for any questions. So Microfluidics is part of the material processing technology or MPT platform under IDEX Health and Science segment. We've been around for about 40 years and we're specialized in produce high shear fluid processors using our unique interaction chamber technology. We're located just outside of Boston, Massachusetts, and we have a global presence. We have sold many units all over the world, both for R&D and production. As I mentioned already, we're part of the IDEX MPT platform. Our technology and processors are used in a variety of different industries for different applications, primarily pharmaceutical industries. Our systems and technologies are used for API manufacturing, vaccine manufacturing, and delivery system manufacturing. We're also used in many other industries, such as chemical industry, food nutraceutical industry, and cosmetic industry. Over the years, we have built a lot of experience in both applications and machine designs, which will help us best help our customers for their development and production. So I want to talk briefly about evolution of vaccines. I want to say that I'm by no means an expert on this subject, and I believe this group of audience also does not need further education. So I will just say a few things here. I think everyone knows that vaccines help our immune system develop antibodies to a specific pathogen and prevent infection from further future exposure. So additional or first generation vaccines use either killed or weakened live whole pathogens. Then move on to the second generation or subunit vaccines. They only use the antigenic parts of the pathogens. One good example of such is the recombinant protein-based vaccine. The latest development or the third generation vaccines directly introduce nucleic acids to achieve in situ production of target antigens. One thing I want to point out here is that both the first and second generation vaccines require long development time, but they both are matured technologies with approved products and all have established facilities for large-scale manufacturing. The new DNA RNA vaccines are novel vaccines which are promising with a faster development and production speed. However, the major issue with them is currently there's no DNA or RNA-based vaccines have been approved. Although there's a few promising COVID-19 vaccine candidates were developed with this technology, and a couple of them even have entered the late stage clinical trials. Among many components required to make a vaccine, I'm going to only talk about three main components here. The first is antigen. Antigens are molecules or molecular structures which induces immune response and hence the production of antibodies. As I mentioned before, they can be either the whole pathogens or antigenic parts. The next is adjuvant. Adjuvants are immune stimulators that can greatly enhance immune response and allow antigen sparing. They have been widely used in both the first and second generation vaccines. Traditional vaccines use alums as adjuvants, but novel adjuvants use oil-in-water emulsions. A good example of that is the squalene-based oil-in-water emulsion, as shown here, and as an example is the MF59 adjuvant developed by Novartis. Another similar adjuvant is the AS03 adjuvant developed by GSK. Both are very well-known adjuvants and have been used in approved flu and pandemic vaccines. Since nucleic acids normally cannot withstand physiological conditions, so a delivery system is often required, which is the third component I'm going to talk about. There are commonly two systems are used as delivery system for genetic materials. The first are viral vectors. One example of such is the adeno-associated viruses. The second group are non-viral vectors, and among them, the lipid-based nanoparticles are the most popular and promising systems. Although microfluidics can be used for manufacturing all these key components, for the purpose of this presentation and the time consideration, the rest of my presentation will only focus on the two major components, specifically the oil-in-water emulsion adjuvant and the lipid-based delivery system. When talking about developing formulation and processes for vaccine components, there are many factors need to be considered. Common challenges and requirements as shown here when generating those nanoparticles, especially for the adjuvant system or the delivery systems. The first and foremost are physical properties, which means your particles need to meet the target properties. One of such is the particle size. The process need to be able to generate particles to meet the desired particle size, and also provide a good or narrow distribution for a number of different benefits, including enhanced stability, be able to penetrate and deliver to the target site, and also can benefit for other adjacent or downstream processes. Once a process and formulation has been chosen, you want to make sure that selected process are robust enough to be able to generate excellent batch-to-batch repeatability, and also future-proof that when you're ready to scale up, you can get a same and guaranteed scale-up results. As I mentioned before, if the process can also benefit downstream processes, that's an added benefit. An example of that is sterile filtration. As we all know that most vaccines are delivered by injecting into body, which means the product needs to be sterilized. An easy and economic way to sterilize vaccine products are through filter sterilization. The membranes used in the sterile filter generally have a rated size of 0.22 microns or 200 nanometers to be able to remove bacteria efficiently. This also means that most of your particles of the products need to be smaller than that. Particles bigger than that can clog the filter, which will lead to an increased filter area, hence the filter cost. Anything will not pass the filter will consider it as a loss of the product. Last but not least, the process needs to be able to meet regulatory requirements, such as be able to comply with cGMP requirements. And many processes also require that the system is capable of running the CIP or SIP processes. Microfluidics technology can actually address all these challenges and requirements from development to production with its unique advantage in achieving process results and guaranteed scalability. By that I mean microfluidizer processor ensure that every milliliters of material gets the same high shear treatment regardless if you're processing just a milliliter batch or running at thousands of liters per hour flow rate. That leads to my first case study to demonstrate the capability of the technology. This study was done at our long-term partner, IDRI, the Infectious Disease Research Institute. IDRI was working on developing an RNA-based vaccine for Zika viruses. As part of this, they need to come up with a formulation and a process to be able to generate the delivery system. And they chose the two-step or two-vial approach. Many of you are familiar with the single-step or one-step approach. In that approach, you generate the nanoparticles and encapsulating your genetic materials at the same time. It's a simplified process and also and often requires use the bottom-up approach and involves solvent. That means the downside of that process is first they have to be able to remove the solvent and then concentrate your product. The major issue is the genetic material usually require a very extremely cold storage condition, usually at -80°C. That means stockpiling a large quantity of this final product may be problematic or even not feasible. So by doing the two-step or two-vial approach, you manufacture empty delivery particles and then mix them with the RNA antigens just prior to the administration. So the lipid nanoparticles are usually much more stable and they don't require that extreme cold storage condition, which means you can stockpiling large quantities and be responsible in a short time for a pandemic situation. So that's why IDRI chose this approach. And they formulated their nanostructure lipid carrier, the NLC, with a hybrid formulation, which comprises both liquid and solid phase lipids to form the core. And another key ingredient here is the use of a cationic lipid, which is the DOTAP, to provide positive surface charges that allow RNA complexing afterwards. Once the formulation with the key ingredients has been determined and IDRI went on to optimize the formulation as well as the processing conditions to be able to achieve and optimize the particle size, RNA loading capacity, efficacy, and also balance between immunogenicity and reactogenicity. And this was all done by top Microfluidizer processor, the 110 M110P system as shown here. This system, as all of our other systems, operates under the same principle as shown in this diagram here. All system has a constant pressure intensifier pump, which pressurizes the incoming sample, usually a crude emulsion or a crude liposome or a crude liquid mixture, to a desired pressure up to 30,000 psi. And under this high pressure, the sample will be forced to pass through a fixed geometry interaction chamber. We do have two different chamber designs, and shown here is only our Y-type chamber, which is the ideal design for the liquid-liquid dispersion, which means in this application. The Y-type chamber has 2 microchannels, so the fluid will be split into 2 streams, and each will pass through the microchannel, subjecting to high shear forces, and then forms an impingement downstream. The impingement leads to extensive turbulent dissipation and micromixing, which all help generating more uniform nanoparticles. All systems are equipped with efficient temperature control, either via a cooling coil or a heat exchanger, to be able to control the temperature as desired output. So after optimizing the formulation and the processes, the results are shown here. As you can see, the optimized formulation is the NLC V2. This nanoparticle has a final particle size around 40 nanometers with excellent RNA loading, up to 100% RNA bonding at a concentration of 10 milligrams per mil. And another data which is not shown here is The nanoparticles provide 100% RNA protection against the RNA's degradation. Then IDRI went on to use these optimized particles and their RNA antigens to do the mass model, to perform a mass model. So you can see at the two graphs shown here down below, the dose as little as 10 nanograms was able to produce a good amount of antibody in the mass, and when challenged 30 days after a single administration, you can see this low dose was able to provide 100% protection and a survival rate. So this study certainly demonstrated the capability of our technology in processing a variety of formulations and produce lipid nanoparticles that meet the desired property requirements. If you're interested in learning more about the method or the results, please either read our published public applications note, which provide a summary of this article, or just go ahead and read the full article. When it's time to consider process scale up, this brings to my second and also the last case study, which is large-scale production of the vaccine adjuvant nanoemulsion. And again, this was done with collaborating with IDRI. The goal is to scale up the production of the IDRI's squalene-based oil and water emulsion as a vaccine adjuvant. The project goal is to scale up the process to be ready for a pandemic response. The objective of the scale up is to be able to manufacture 50 million total doses of adjuvant within 3 months. So after calculation, factoring a few assumptions and manufacturing parameters, IDRI come up with to be able to meet this objective, they need to be able to manufacture 1 million doses daily. Obviously, by scaling up, you need to achieve the same performance criteria. In this case, the primary goal is make sure the particle size stay within 100 nanometer range, and also other parameters as the oil and excipient concentrations, visual appearance, and pH, and etc. Should also match with the lab scale results. Lastly, finally, the large-scale batch needs to be produced at IDRI's cGMP facility with the cGMP equipment. So all this data was just recently published in a special COVID-19 issue of Pharmaceuticals. I'm going to show you some results here. First, I want to show you what we achieved with our pilot scale system using our M110EH machine here. This system provides up to about a third of a liter per minute flow rate. The results shown here are actually optimized the process and formulation. Their original formulation has a very low oil concentration and require many passes. I mean, at least more than 10 passes to achieve desired particle size. You can read the details from the paper, but what I'm showing you here are the optimized formulation and process. You can see with the optimization, you only need either 2 or 3 passes to be able to achieve the desired particle size with an excellent narrow distribution. Then we're ready for scaling up this process. So the way we scale up is not like many other methods or technologies. Other technologies usually scale up by increasing their geometry or size. We don't do that. We don't give you or use a larger microchannel. Instead, we actually use identical microchannels, and we use multiple of them and align them in a parallel fashion. By doing this, we ensure the physics happen in each channel will be at exactly the same, which means as long as you maintain your process conditions, you will achieve exact same processing results from each microchannel, hence the overall scale-up results. Now, look at the scale-up results here. Remember, this is what we achieved on a pilot scale, and when we run the same formulation and same process on the production machine. In this case is our M7250 enhanced machine. The machine is capable of delivering about 5 liters per minute at max pressure of 30,000 psi, which translates to about 300 liters per hour as a throughput. You can see there's only a slight difference after the first pass. Actually, the production machine made it a little bit better after first pass, but after the second and the third pass, the results are almost identical because you can see the curves are all overlapping with each other. That demonstrated our excellent scalability from small scale to large scale. Then IDRI did additional optimization of adjacent processes, including the upstream premixing step and downstream filtration step. So after overall process optimization, they found they were able to improve the process efficiency and increase the production capacity to 5 million doses per day. This is like 5 times higher than their original target, which is 1 million doses per day. And all this 5 million doses can be done with a 200-liter batch and total processing time less than 8 hours. So the total processing time includes starting from scratch, the premixing and the microfluidizer processors processing time, and also the downstream filtration time. All this can be done in just a single 8-hour shift. That means the process has been greatly improved, and they were actually being able to produce more doses per day than they have originally targeted. Now, after both case study, I want to show you our diverse product portfolios. We do offer a range of different benchtop units, as well as a couple of pilot systems and custom production units. So we can process as little as a few mLs as batch size, all the way to hundreds or thousands of liters in batch size. The final thing I want to point out here is starting from this benchtop unit here, they all have the cGMP version, which means you can use the small benchtop cGMP model for small clinical batch production, and then move on to for larger clinical batch production, eventually to commercial production. All with the same results. With that, I'd like to conclude my presentation, and I would like to thank everyone for your attention. I'm happy to answer any questions that you may have. If I don't have enough time to answer the question, or if you're interested in learning more about our technology and other applications, we do have a virtual booth here at this conference, and please make sure you visit our booth, and we'll be happy to address your any additional questions or issues. Thank you.